Alright, what we're going to talk about are contact plates and how bacteria are ubiquitous. So here's a contact plate. This plate has a grid to help you count colonies if you're looking at quantifying it. Contact plates are special because they have a very small lip on the side. When you pour the agar in, the agar upraises on the top of this so that all you have to do with the agar is take this, place it down, lift it up, and you get bacteria. We have here contact plates where we incubated them for 48 hours from a public toilet, the palm of your hand, a handrail on stairs, a can, computer mouse, yoga mat, elevator button, chip bag, and makeup sponge. Now we're going to look at each one of these here. I'm going to pull this over. We can look at our makeup sponge. All of these are bacteria on here. Each circle then is a colony. The colony was formed by a single bacterium which then duplicated into the whole colony. The original bacteria were called colony forming units, or CFUs. Here's a nice spreading colony here, and then here are your normal circular colonies. Let's look at our chip bag here. Okay, very interesting. We can definitely see different morphology of those colonies. There is an elevator button. Okay. Interesting, a lot of spreading colonies on that one. A yoga mat. Our computer mouse. This one's interesting. There on the right, all of that white powdery stuff is a fungus. So big old fungus. You can see all those spores on top of that fungus there. Okay, the other items in there are bacteria. Different types of colonies on that one. Here is a can. A lot of different colonies on that one. Kind of cool ones. The handrail of stairs. Okay, really cool. The palm of somebody's hand. Tons of bacteria on that. And finally, a public toilet. And we don't see any bacteria hardly at all because public toilets get disinfected all the time. Okay, so here is a contact plate. This contact plate is PDA. This then was smashed against the keyboard of a computer and then was incubated. And here we can see that we have growing on this both fungi and bacteria. Fungi are fuzzy, like this big one is clearly a nice fuzzy fungus. Other ones in here you can see are fuzzy, and then we can see our slimy ones, which are bacteria. So you can go ahead and look at those, make your estimates, and record your data. Now, I want to show you how to make a smear preparation from a colony on a contact plate. This contact plate was used to press against the computer keyboard, and we can see there's a lot of bacteria, and even up here there's a little bit of a fungus. What we want to do then is choose one of these colonies, move it to a slide, stain it so that we can view it underneath the microscope. The first step then, I want to take some DI water and put a teeny tiny drop of DI water on my microscope slide. Okay, that allows everything to spread around nice and easily. I'm going to take my loop, do a flame the loop. Okay, we'll let it cool down just a speck. I'll open this up. You notice I'm staying underneath my sterile area on here. Touch it to a neutral spot to cool it off, and then I'm just going to grab a single colony right here. Doesn't take a lot. We'll just get a little bit of that colony. Put this lid back on, then I'm going to stir it around in that little drop of water. Nice and big so everything is on that drop of water. I'm going to take my loop, make sure that I sterilize the end of my loop so that everything is killed and sterile on that loop. Now I have to wait a few minutes for this drop of water to evaporate before I can heat fix it. So definitely you do not want a large drop of water, or else you'll be waiting a long time for that to evaporate. Okay, so a few minutes have passed and this now is dry, ready to heat fix it. So I'm going to move this out of the way. We'll move our plate out of the way. We'll keep our slide right here. And we will then add some DI water and we will add a staining tray. Okay. The next thing I want to do is I want to heat fix this. And so I'm going to take my slide and I'm going to attach a test tube clamp to it. I'm going to angle this up so that I get a good angle on it, and I'm going to just pass it over the top of the flame, 1/1000, 1/1000. Now this end is going to be hot, so I've got to be careful here. So now we just have to make sure that we gently get this slide onto the staining tray. Okay. And now that the slide is on the staining tray, what I'm going to do is just cover the smear with methylene blue. And then we're going to time it for 1 minute and leave that for 1 minute. Now 1 minute has passed, so I just basically want to take some DI water here. We'll take it, angle that down so I don't get in any on me, and we'll then go ahead and get all of that water off. And I'll take some Chem wipes and I will blot it off gently with Chem wipes. Alright, so now I've blotted it dry with Chem wipes. This is our slide. It's all ready to go and be used under the microscope. I'm going to repeat that process on the contact plate 2 more times, making a total of 3 slides, and we'll view all 3 of these slides underneath the microscope. Alright, so here we have our smear preparation of bacteria taken from a colony that was pressed against a keyboard on a contact plate. The smear preparation was then stained with methylene blue, and these are the results. This is for X. I'll switch up to our 10x magnification, refocus. We will then switch up to the 40x magnification and refocus. Okay, and finally then we will go all the way up to the 100x objective. We'll add a drop of oil to the slide. We'll swing in the 100x objective. We will increase the light and focus. Alright, so these are nice, easy to see. You can get the arrangement and you can get the shape of these bacteria real easily. Okay. For size, assume that the diagonal is the diameter of field of view. Now I'm going to go over here to another smear that is from a different colony. Okay. That's part of the same one, so we'll move to a different colony on here. There we go. So here's the other colony. So I did 2 colonies on this one. Here is my second colony. These bacteria are also taken from the same contact plate. And we can see here that their shapes and arrangements are different. Alright, so here is our third smear. This is under 10x. We'll shoot this up under 40x and refocus. Okay, I'll then go halfway between the 40x and the 100x. Put a drop of oil right on the slide. We'll swing in our 100x refocus, increase that light so we can increase that resolution so we can see these. And these are nice and easy to see as well. So we can see those arrangements, and you can see those shapes in there on these guys. Alright, there we go. Alright, so now we're ready to work with our other contact plate. This is the PDA plate, which favors fungus. This was also put on the same computer keyboard, and we can see here that instead of bacteria, we have a lot of fungal growth on there. Right? Funguses are fuzzy, bacteria then are more slimy. Right? So what I want to do is I basically want to make a wet mount. So I'm going to put a drop of water right on top of this microscope slide. There'll be a little bit more water than what we did for the bacteria because we were making a wet mount this time. We'll heat this up. I'll heat up my other set of tweezers here so that our tweezers are ready to go. We'll take the lid off the plate. We will go in and actually grab something here that has a lot of fungus on it. Cool those tweezers off a little bit. And we will grab some good hyphae from that. And then we'll get the hyphae down inside the wet mount. Okay, I'm going to reflame my tweezers here. Make sure everything's off of that. I'll put the lid back on, and I'm going to take a cover slip. Well, first I'm going to take a drop of lactophenol cotton blue. We'll put the lactophenol cotton blue on the slide. We will then take a cover slip, put that over our slide, and now we're ready to view that underneath the microscope. Alright, I'm going to show you the slide of the fungus sample. The fungus sample was taken from the contact plate, from the keyboard, and was stained with lactophenol cotton blue. This is the 4x objective. I'm now going to shoot in the 10x objective, and we'll focus under the 10x objective. We will now swing in the 40x objective. We'll increase the light, and we'll move to the edge where we can actually see some of these structures here. Okay, and here we can see all of these string-like things are hyphae. At the tips of a lot of these string-like things we have spores. The little round things at the tips of the hyphae are called spores. In this case, the spores look like they're in chains, right? But here we have hyphae with spores.